Okay, so hello everyone. My name is Mikayla. I was working with bees this summer instead of plants, but it's okay. I'm going to be talking about Crithidia bombi. So let's get started. Okay, so just for a little quick introduction, so bees are really important to the pollinator population here on Earth. There's hundreds of species known, and some of these species can get infected by parasites, one of which that we've been working with this summer is the bumblebee. And although not a big threat, parasites still negatively affect bees on a daily basis. They're very microscopic, so they're not like right on their back or anything. They're right inside themselves. And they're known to also just co-evolve in general with their hosts. And the reason why this is important to really study is because bees are important to the agricultural food production that we humans provide into the environment. So they help with pollination of fruits, plants, and vegetables. And when we have parasites within these bees, it causes for potential spillover events. So if one bee happens to travel to a flower, maybe excretes on this flower, another bee from another population happens to go to that same flower, somehow ingest that parasite, it then becomes a potential for the next population to become infected. So just a little bit of a background. So Crithidia bombi is a parasite I was working with this summer. It's a gut parasite that's well known to infect the bombus genus, otherwise known as the bumblebee. One of the things that was noticed is that it can cause high mortality, especially to the queen, which results in a smaller colony size and lower reproduction rates. It also increases starvation and reduces the foraging for the bees to go out and collect pollen or nectar. So as I said before, these parasites are often spread through bee fecal matter on surfaces like flower petals, or again, spillover events can occur where a bee can transmit to another bee. And the objectives, before I move on to that actually. So I have a picture here showing the microscope, and in this little red circle is a little cell of Crithidia bombi. I wish I had put a video, but normally they're really sporadic. They like to move around a lot, swim very fast, very rigid in their motions. But the objectives here were to observe and manipulate which bees can potentially get this parasite and whether or not there's some form or way for some of these bees to have infection replication. So my hypotheses for this were, I had two experimental topics. So I did manipulative and observational. So the first hypothesis is that some bees that may potentially be closely related to the bombus will become infected with Crithidia bombi. And the reason why I thought this was, well, if we know that in general, some parasites can have some replication or some co-evolution with their hosts, well, who's to say Crithidia bombi might not have some evidence to support that? And for the observational portion of this experiment, we did screenings. So I thought maybe Crithidia bombi will be present in other species or genera than the bombus genus. And the reason for that is just, well, if maybe bees who happen to visit these flowers can potentially get it. So my screening methods. So over the summer, we caught a lot of bees. Here's a little bumblebee here sitting on a little tube of sucrose. And we'd go to various sites around Ithaca. So we went to, just to name a few, the Ithaca Children's Garden, the Cornell Botanical Gardens on the Pollinator Path, and the Van Horn Park in downtown Ithaca. And to do this, we use nets and put them in some little tubes and then transfer them in the lab in these little salsa cups. So they were, mainly labeled with the place that they were from, the date, and also an ID number. Of course, as I mentioned before, there's a little sucrose container that the bee's hugging on. And once we were ready to dissect these bees, as these parasites are inside their guts, we had to knock them out with carbon dioxide, remove the head from their body, and then go in through the abdomen where their stinger is and just pull out the gut. After we did that, we then put some PBS solution in some vials, depending on their size, crushed the gut up with the pestle, waited about 2 hours for the gut and the PBS to separate from each other, and then took about 10 microliters and placed it on a hemocytometer and screened it on a microscope. And what we were able to accomplish was we did about 500 bees, no, over 500 bees. And this table here is just showing you general information of the families that we were able to dissect. The number of the samples and the proportion positive is essentially, well, how many bees out of the total number of samples were positive. So as you can see, like Adrianidae had about 8%. And the mean number of Crithidia cells in the positive samples is just essentially, out of all of the positive bees, what was the mean number of cell count? So again, the Adrianidae family had the most, with about over 200,000. So this is a little bit more of a specific graph or table that we had. And one thing that I'd like to point out is all the categories are the same, but the bombus, which is right here, only had about 48 and 2% were positive. But the most important thing is that right here, you can see that there's a really high number of mean Crithidia cells, which just goes to show that even though we had like one bee with Crithidia, it was really, really prevalent within their system, and that it's very associated with this parasite. So then on to our inoculation methods. So it was very similar to the screening method. However, instead of just dissecting this bee, the moment we got it, we would take some lab infected bumblebees that we had already had Crithidia bombi in, created a gut sucrose solution for them to eat, and knocked out a wild bee. So we have a little bee here with a micropipette. Inside of it is our gut sucrose solution. Normally their tongue is sticking out, so when they're waking up, they're just tempted to eat this as they're waking up. This is nice and sweet, and they think it's something delicious. And after we did that, it was dissected after 7 days, and these are our results. So and before I go on, some of this data, as you can see on the bottom, was from a summer 2020 inoculation. Sadly, we didn't have very many bees survive from our experience, so we used some unanalyzed data and combined it with ours. So on the left here, we have bee family, and on the y-axis, you can see this is a proportion of Crithidia cells relative to the inoculum dose that we gave them. So the Collectidae family has a really big amount of replication. So I didn't mention this yet, but this red line here is essentially just showing you a threshold. So if any bee were to happen to be positive and show that they had Crithidia after being inoculated, and they were above this line, that just shows that, well, Crithidia are replicated within their system and they are now considered infected. So again, the Collectidae had a very high number of infected bees. So also with the Aphidae family. And to get a little bit more specific, on the right, we have a bee genus graph. And here, our biggest contender for being infected is the Hylaeus. So here you can see it's a really long column of just positivity rates. We also have a few bees from Serotina and one bombus that was infected, which is quite interesting. But if any bee fell below this line and actually had some Crithidia within its system, it didn't replicate, but it's still within the system. So it can still just pass through and not really infect this bee at all. So the question then is, well, why is that? Does evolution play a role in this? And if you look here, as I mentioned before, the Aphidae family is way up here, and the Collectidae family that had a lot of replication is way down here. This is our Hylaeus bee, or at least a species of it. And the most recent common ancestor for this bee is right over here. So at least with the data that we have now, evolution doesn't really seem to support, or our hypothesis is not supported that really evolution may have a role, at least with this data set. So just to give a quick summary of our data, a nice little background of bumblebees. Crithidia bombi was found in numerous genera in the Ithaca community. And the data suggests that, well, bombus isn't the only genus to have Crithidia bombi in the wild, and it's certainly prevalent. And inoculations were successful enough to show some data that relatively shows that these two families, Aphidae and Collectidae, can also have some replication within their system, even though they aren't closely related to each other. Sadly, my hypothesis wasn't supported by that. And now, where will the project go next? So The next question, or at least the next step is, well, where is Crithidia mainly prevalent in? Is it located in environments where there's a lot of stress, or is it located in places where there's not a whole lot? So if there's like an urban environment where maybe there's patches of flower scattered around the city, you know, are these bees that are frequently visiting these flowers, is it higher infection rates, or is it where there's a lot of flowers, a lot of opportunities and patches nearby that it happens to spread to even more populations that are congested within this one area? The other question then too is, if I can go back really quickly to this table here, you can see that Hylaeus, which was found to have really high replication rates in our inoculation data, has had 0 positives in the wild, even though here clearly there's a lot of data that suggests that they can have Crithidia. So a future question for this is then, well, why is this? Is there a reason why Hylaeus didn't have any in the wild, at least in the sites that we investigated? And maybe there's some pathways that, like, do they puke when they go to their nest, like supply food for their young? But yes. Now I'd like to give some acknowledgements. So I'd like to thank Scott and Paige, hi Paige, for allowing me the opportunity to work here. Emma and Lauren for the inoculation data from the summer of 2020. Wei Hao for our pretty R graph that you just saw. And Michelle, Savannah, and Jeremy, our other interns as well as workers in the lab. So thank you. Hmm? Yeah, so we ID'd like pretty much every single bee. So any bee that we found to be positive, we ID'd it to the point where it was to their species specifically. And all the bees that we had inoculated and the ones that we have found in the wild, except for maybe like 1 or 2, were all bombus impatiens, which is just a common bumblebee essentially that we have around here all over the place.
How many bees lost their lives through your summer research?
Oh my goodness. Honestly, I killed a lot of bees this summer, I'm not gonna lie. But we did actually catch over 1,000, but sadly, a lot of them ended up dying to natural causes. Sometimes their heads got squished in the containers. So it was kind of sad when that happened, although they met their demise early. So I guess that kind of saved them in the long run. Oh, yeah. So, okay, for all the wild bees that we caught, we would use nets. We didn't have any protective gear, so it was just like we go in right away. We had these little vials with little tubes that had holes at the top. If they weren't big enough, the bee won't escape, but sometimes they would as well. And we just had to be really careful not to get stung. You just like grab the net and the bee's up here. You just sneak the tube up there and try to grab it. We had some carpenter bees that were really hard to catch because they're just so big. They're absolutely ridiculous.